This is improved Neubauer chamber. It was invented by Kramer and it was modified by Neubauer. This is a single solid heavy glass slide which has 3 parallel platforms. You can see here, this is one, this is second one, and this is third one. This is the central, this one is the central platform. And these 3 platforms are separated by 2 gutters. These are the 2 gutters you can see here. These are the 3 platforms. They are separated by these 2 gutters, and here also centrally also this gutter is present. So the gutter gives H shape. So it is a set of gutter in the shape of H. Now the central platform or the floor piece, this one, this part, is wider and it is 0.1 millimeter lower than the lateral platform. So these 2 lateral platforms are 0.1 millimeter higher than this central platform. What is the use of this? Whenever we are placing cover slip on this Neubauer chamber like this, here when we are placing cover slip on this central part, so the depth of the area in which we count either RBC or WBC in different counting chambers we'll discuss. So the volume becomes, or depth of that volume becomes 0.1 millimeter. Now, identically, ruled areas are known as counting grids. These are the counting grids you can see. If you can see it carefully, visualize this, you can see there are counting grids placed on this central platform. These counting grids, they measure about 9 millimeter square area. 9 millimeter means, I'll show you the diagram. This is 9 millimeters square surface area. 9 millimeter means 3 millimeter by 3 millimeter. So 3 millimeter length, 3 millimeter breadth. Okay? And here further, this counting grid, which is further divided into 3 1 millimeter square areas. This is 1 millimeter by 1 millimeter. This is again also 1 millimeter. This is 1 millimeter. Okay? Of this 9 squares, 4 corner squares, they are employed for the counting of WBC. So they are known as WBC counting areas or WBC counting squares. These 4 squares, here you can see this 1, 2, 3 and 4. These 4 squares are further divided into 16 small squares. You can see here 1, 2, 3, 4 and these are 4. 16 small square, or you can say medium size square, because this central one are smallest one. Okay? Now this centrally placed densely packed squares, that is for RBC. So this is known as RBC counting area or RBC counting square. This central square, again the size is 1 millimeter by 1 millimeter, and which is further divided into 25 medium size square. You can see here. This 25 medium size squares, they are separated by triple lines. These are the triple lines, they are separate. Okay? So this 25 medium size squares are available, and this 25 medium size squares are further divided into 16 small sized squares. You can see here in this 25 small squares, we again have 16 smallest squares. Okay? Now we discuss about the volume of each and every square, WBC counting square as well as RBC counting square. Let us start with the WBC counting square. WBC counting square, as we have discussed, the length and the breadth, that is 1 millimeter, and depth is, as we have discussed, 0.1 millimeter or 1 by 10. So the total area would be 1 millimeter multiplied by 1 millimeter multiplied by 0.1 millimeter. So it becomes 0.1 millimeter cube. Okay? And what about the area of this small square? That becomes 1 by 4, because the whole is 1. So this is 1 by 4. So 1 by 4 multiplied by 1 by 4 multiplied by 1 by 10. So it would be 1 by 160 millimeter cube. Now we discuss about the RBC counting square. In the RBC counting square, again this is 1 millimeter, this again is 1 millimeter, and the depth is 0.1 millimeter. So what about the area of this medium size square? That is 1 by 5. This is also 1 by 5. So the volume is 1 by 5 multiplied by 1 by 5 multiplied by 1 by 10. So the volume would be 1 by 250 millimeter cube. And here for RBC counting, what we do is we count RBCs in 4 corner squares and the central square. So whatever volume we are getting, we have to multiply this with 5. So our volume is 1 by 50 millimeter cube. Now let us focus the same chamber in the microscope and let us discuss more about the chamber. Now we are focusing the counting chamber. This is the lowest magnification, 4x. We are not supposed to count RBC or we are not supposed to count WBC in this x. This is just to visualize this chamber. You can see here, as we have seen in the picture, these are WBC counting squares. This is one, this is second. So four corner squares are there which we have seen. This is third one and this is the fourth one, and this central one is for RBC counting. So this is RBC counting square and this four are WBC counting square. So first of all, whenever you are placing Neubauer's chamber, you have to examine this Neubauer's chamber in low x. Here you can see this is WBC counting square, which can be seen under 10x. This is one WBC counting square, this is the second one, and this is third, and this is fourth one. So four corner squares are there. This center one having triple lines. You can see these are the triple lines, and the central area which is further divided into 25 medium sized squares. I'll show you here. You can see this is the central part for RBC counting. Now we are visualizing the Neubauer's chamber under high power. In high power, here I have focused the RBC counting square. As we have discussed that RBC counting square has 25 medium sized squares. So this is one of this 25 medium sized square which is bounded by triple lines. And this medium sized square again has 16 small size squares inside that medium size square. So you can see here this is the RBC counting square in high power. Now this is new improved Neubauer's chamber. How it differs from old one? That is, in old Neubauer's chamber, the size of the central area, that is same 1 by 1 millimeter square, but it is divided into 16 groups, 16 medium sized square, and these 16 squares, they are separated by double lines. Here instead of 16 squares, in improved chamber there are 25 medium sized squares, you can see here, and they are bounded by triple lines. So this is all about improved Neubauer's chamber. Thank you.